outside, while smooth does not. Functionally, what are some differences between rough and smooth? Rough ER functions? Protein and phosphorus. Very good. Smooth ER? Cholesterol steroids, detoxifies, and then we talk about mention also helps you break down collagen. We mentioned in lab that Golgi is mainly for packaging, so it receives things, chemicals, these products from the endoplasmic reticulum, it will modify them, it will concentrate them, and then they package them and send them to different places, right? So they can send them to become part of the membrane, right? What's the most common molecule in our plasma membrane, in our cell membrane? Phospholipids. So it comes from the, where is phospholipids producing it? Raphe. So it comes from there. Right? Proteins come from the raphe, get put together and make them large and become part of them. We can also secrete them to the outside of the cell. That's the secretory vesicles. Okay? To the outside of the cell. So for example, mucus, for example, is inside the cell, they're packaged, and then they are sent out. Right? Hormones, same way you talk about steroid hormones, they're produced within the cell from smooth ER, they'll go to Golgi, they'll be put into a vesicle, and then they'll go out of the cell, and of course out of the organ too. Okay? While others stay within. An example is lysosomes. What are the contents of lysosomes? Enzymes, right? What are enzymes made mostly of? Proteins, right? So they also come from our gut. So they could be, that's an example where it could stay within the cell, right? These proteins can stay. So either they stay within the cell, they're secreted out of the cell, or they become part of the membrane itself. Okay. What else do we want to talk about here? This is just expanding on what I just said. So we talk about secretory, or inside, like lysosomes, or becoming part of the membrane itself. Next, lysosomes. So we mentioned they have digestive enzymes, so what's the main function of lysosomes? Digest, right? They break down things. Okay. So we're going to talk a little bit about autophagy, autolysis. Autolysis, they'll break down the whole cells. For example, if it's a whole cell or infected cell, if they receive the right trigger, the lysosome just burst open and they just digest the cell from inside out. Okay. That's autolysis. And then we have autophagy. I did not put down that just eats up the organelles itself, right? So they are enzymes that can break down other things that are brought into the cell, like macrophages will swallow up bacteria, or bring it in, lysosomes combines to it, and then they destroy those cells, so break down the cells. Peroxisomes, what did they do? Yeah, free radicals. Free radicals, if we have too much of them, they will actually cause mutations in our DNA and can lead to cancer, right? How do we define cancer? How do we define cancers? What are cancer cells?
Mutated cells.
They come from mutations, yep. Uncontrolled replicate cell division. Does it have to spread to other organs to be considered cancerous?
Yes.
No, it doesn't. Okay. We'll talk about benign versus malignant. It's the malignant that does spread. So cancer itself does not need to spread, but it's the uncontrolled cell division. So this can lead to a DNA change or mutation, which can cause. We'll talk about a little bit about the cell division. We'll talk about this different part. Mention about checkpoints. These kind of bypass checkpoints, they're not being destroyed, even though they're not regular cells. Mitochondria. So we talked about what was the main function of mitochondria? Produce ATP under what conditions? Oxygen is present. Right? We'll add a little more detail. We'll come back to it believe it was chapter 4, when we do the digestive, we'll talk a little bit about it. It is double membrane, okay, and it's within the second itself that the enzymes are going to be found. So a quick drawing, outer nice and smooth, the inner membrane has folds. What is the reason for having folds? Increase the surface area, like we mentioned. We talked about microvilli, we mentioned it. So this is the mitochondrial matrix, I'm coloring red. It's like the cytoplasm of these mitochondria, which contains the enzymes for cellular respiration, which we'll talk about. Double membrane, we have outer nice and smooth, inner one, the cristae are those folds, and then the matrix will be the red in this one, which is this kind of like a cytoplasmic equivalent of so inside the cell. This contains the enzymes for required for this function as a cellular respiration. It's aerobic. Why is it aerobic? There's no oxygen, mitochondria. The only additional features here is the structure. So outer membrane nice and smooth, inner membrane has folds. Those folds are called cristae. The mitochondrial matrix is the cytoplasm equivalent, the liquid portion within the mitochondria where the enzymes are present for that cellular respiration. Some of this is revision, so we talked about nucleus, right? What's the function of the nucleus? Yeah, it contains our genetic material, right? Generally, one nucleus per cell. Do we know of any exceptions? Have we heard any cells having multiple nuclei or no nuclei? They still have one nucleus. Generally one, while they're divided, it could be considered two for a few seconds. Any cell could be. Cells the same. What about blood cells?
They don't have any nucleus?
All blood cells?
Red blood cells.
The red blood cells, mature red blood cells do not have white, while the white blood cells have a copper nucleus, right? But the red blood cells do not have a nucleus. Any cells that you know? Neurons do have just one nucleus. The reason we may see other nuclei are the cells surrounding the axon, so it's not part of the neuron. Our skeletal muscles, they actually are multiple nuclei. Tens, even 100 nuclei per cell. So most contain one. We give a couple of examples that are not one. Okay, they do contain genetic material. Generally in the center, but not exclusively inside. And in lab today we looked at adipose connective tissue. The cells is filled with fat, adipose, and then the nucleus actually pushed inside. So we have a nuclear envelope. Who recall what the nuclear envelope is? Yeah, so it's the boundary of our nucleus. So this nuclear envelope controls what goes in. Now, these nuclear envelopes actually have bigger openings, right? In our plasma membrane, there are some. It is selective permeable, but smaller particles which can pass here. Even large particles like RNA can pass through our membrane, our nuclear envelope. Right? So separate out the cytoplasm from the rest. What is this nucleolus? What was it made of? What's it function? All the RNAs and proteins as well. That's why it's darker. Yeah. This whole thing's a nucleus, by the way. This is an electron microscope image. This whole thing's a nucleus. You can kind of see this could be the endoplasmic reticulum. And then you see a darker spot, that's our nucleolus. What is the function specific to nucleolus? Produces the ribosomal RNA. The nucleolus produces ribosomal RNA. So nuclear pores are large openings. Here's a nice nuclear pore. And again, this is three-dimensional. You only see one pore there on the 2D, but there are multiple of them. Lots of openings, so hence bigger molecules can pass through into and out of the nucleus. Okay, becoming normal ones as well as larger molecules. Proteins are molecules through these nuclear pores. These are opening in the nuclear membrane. We mentioned about the nucleolus. It is DNA, RNA, proteins. It produces, synthesizes ribosomal RNA. While chromatin, I know we're going to talk about now a little more genetic terminology. When we say chromosomes in genetics, we mean a specific structure of our genetic material. Okay? While in a non-dividing cell, chromosomes or DNA are in what's called the chromatin. By the way, does anyone know how long, if we stretch out how long is DNA, how long it is? It is very long, right? How big are cells? Microscopic, yeah, less than 1 millimeter, right? Generally in the hundreds, maybe tens to hundreds of micrometers. How long is long for DNA? Maybe up to 6 feet long. The longest one stretched out. Right? So we have to pack it into a nucleus. Almost we can even virtually draw it, right? We have to pack it into that nucleus. So it is packed, but we have even more packed. That's why this is called less condensed form of DNA. So in a non-dividing cell, our DNA are in this state of chromatin. Okay? Because we're going to talk about chromosome and then another one called chromatid with a D. Okay? These are terms that are all kind of describing similar things or similar parts of the same thing. Okay? So less condensed form of DNA is called the chromatin. In a non-dividing cell, this is how our DNA is. Right? Did I put the picture? Yes, I did. So here would be double helix. Okay? How we form chromatin is we wrap them around protein molecules. They're called histones. We wrap them, and then you don't even see where it starts and ends. Right? So here you can see, but they're condensed form. They are called chromatin. While when they divide, even under a light microscope, you can actually start to see the other chromosomes. You can see the start and the chromosomes. Okay? That would be the chromosomes. Highly condensed form of DNA. Okay? So just to introduce this. So, let's talk about how many chromosomes do we have. It's 23 pairs. We have 46 chromosomes. Right? We got 23 from the sperm, biological father, and 23 from the ovum, the biological mother's the gamete. Right? They combined of each one of us. Okay? The 23, 22 are very similar. Only the 23rd one or the last one, we call them sex chromosomes. You might have heard of X and Y. That can be different. Others all, the lengths are pretty much the same. The points, we'll talk about this some point. That's called the centromere, like this narrowed area, is about the same length, same position, and we also carry similar DNAs on each of them. Okay? So they are paired. That's why we talk about. I think I mentioned about dominant and recessive. Did I mention that in the class? Okay. So we'll talk a little bit about that too. Okay. Anyway, so first of all, during non-dividing, DNA is wrapped around proteins called histones. They are in a condensed form, but we have even more highly condensed. So this form we say is less condensed. Okay, that's the chromatin. Okay, And by the way, we talk about genes, right? Genes is what translates proteins, that's what gives us different distinctions in our faces or body, right? But how much of our DNA is occupied as genes? Is it 100% all of our chromosomes genes? All of our DNA is genes? So you know? It's only a small part. About 2% of the chromosome, right? The rest can be just placeholders or can dictate, promoters and so on can dictate how genes are expressed. Right? But just note, it's only about 2% of all our DNA molecules contains really information to translate to a protein. So the genetic code talking about proteins. So DNA has to lead to proteins. Okay. How many amino acids do we have? You remember? Lower than 4. There's 20 different amino acids, right? So somehow the DNA information has to translate to one of those amino acids, right? So we only have, when we talk about nucleotides, what was a nucleotide? That is the first part I'm hoping you got. Second part is, what's a nucleotide? It's the monomer of nucleic acids, right? It's the monomer of nucleic acids. So they have a couple of things in common. In a DNA molecule, they're going to have deoxyribose and they're going to have phosphate. Okay. Then they have a nitrogen containing, it's called nitrogen base. That one can be different. We have only 4 different types. So somehow this 4 different types has to be able to give the information for these 20 different amino acids. Okay? We can't do that with one and one. Okay? Because we don't have 20 different nucleotides. If we do 2, that only gives us 4, 2 to the power 4 is only 8. Okay, so only 8. So we actually have 3 of those nucleotides. That's why it's called the triplet codon, triplets. Okay, 3 of those nucleotides is going to code for an amino acid. Then you're going to say, okay, there's 3. How much is that? That can give you actually 64 combinations, by the way. There's going to be some that code for the same amino acid, which is why some mutations, nothing is different. Codes for the same. Okay? Just note that as well. So, again, we don't want to go too detailed for us, but we just need to recognize some things. But I do want to recognize also, as I mentioned, gene. That's our hereditary information. That's what translates to proteins. Only 2% of our DNA is genes. Some of them mentioned it's just placeholders. Some are what's called the promoter. It's kind of like saying, okay, this sequence is the gene, so just before that, there's going to be certain sequence saying this is the start of the gene, kind of giving us the indication where it starts. And at the end too, there's actually a terminating code sequence as well. Say, okay, this is the end of the gene. So that also takes up some. Okay? So understand that too. There's a promoter and a stop signal. Places where they start and happen, because they go on. I mentioned DNA, only a small person's gene. Oh, that 2% I mentioned, it's all of the genes together. It's not that the first 2% is and nothing else. It's actually piece here, piece there, and so on. Okay, so it's not continuous either. So we have to be able to find those points. So, triplet codon. 3 nitrogenous bases will identify a single amino acid. Multiple nitrogen bases, and the nitrogen combinations can identify one. Okay? There are some, which are about 8 different codes that can identify the same amino acid. For us, we just need to recognize that 3. Okay? Protein synthesis. It's a 2-step process. Big picture first. We go from gene, leave a space here for now, and then we want to get to a polypeptide. Okay, that's the that's what is a polypeptide? Polymer of proteins, right? So you want to get there. So from gene, we actually will first form the mRNA. Okay? This process by which we use the information on our gene to form mRNA is transcription. While I'm saying this word out, I'll tell you about HIV virus, right? What's the HIV virus? What does it stand for? Human immunodeficiency virus. It has an enzyme called reverse transcriptase. This virus is actually an mRNA. It's got an enzyme that it can put this mRNA into our gene. Okay? Which is what the transcriptase universe talk about medications to treat HIV. They'll talk about reverse transcriptase inhibitors because we don't want the HIV virus to infect basically our DNA. Okay? And then it uses our DNA to multiply. We can have cancers from HIV. If you haven't heard, it's not just the immunodeficiency. We actually have certain types of cancers that can develop because of this capability. Okay? So transcription is changing the information of getting the information from gene to information sequence in the nucleotides in an mRNA. Translation uses this information to sequence out the amino acids in the peptides. Okay? That's translation. By which we use information on our mRNA to find the polypeptide. What did we say? Which one of the organelles produces proteins? Rough ER. What part of the rough ER produces proteins? Ribosomes. It's actually the ribosomes that read the mRNA and put the amino acid in. And we just put it in. So it's protein synthesis transcription first, where we convert or where we use the information in the gene to make an mRNA. Okay. Translation, we use the information in the mRNA to make a polypeptide. Okay. So the process of transcription is to make formation of a messenger. That's the mRNA. The enzyme here that does that is RNA polymerase enzyme. Okay, RNA polymerase enzyme makes that. We didn't go into the complementary nature, so I'm not going to stress about it. Basically, we have to somehow match the information to the information. Okay, just so it's admin and we didn't go into the developers course. But do recognize, after we form the mRNA, 3 of these nucleotides is going to be equal or give us information for 1 amino acid. Triplet codes for 1 amino acid. Okay? Here's how we call it. While translation, we're going to use the information here to form our polypeptide, the sequence of amino acids. Okay? And the sequence is determined by what sequence of those triplets we have. Okay? There's a certain triplet for one. Again, I'm trying to give us just enough information. We are required to know the basics of this. We go into genetics, I'll talk about how it starts, how the ribosomes truly reads it. Okay? But we just need to know it happens. This is the big picture, right? mRNA information here is translated to polypeptide. It's 3 nucleotides here equals to 1 amino acid. Okay? And it's the ribosome that does the translation. Okay? So here we can add by RNA polymerase. Translation by ribosomes, but also with the help from tRNA. Okay, tRNA basically brings the amino acid to this kind of like a conveyor belt. They just bring the amino acid, give it, and then they'll go grab more amino acids. Okay. That's the big picture of protein synthesis. We go from information on the DNA, a small portion from the gene, we make an mRNA molecule. This process is called transcription. This process involves an enzyme. Mainly, there are other enzymes too, but this is the main enzyme, RNA polymerase enzyme. We produce mRNA. Then mRNA's information is used to translate to the amino acid sequence. That is performed by our ribosomes with the help of tRNA, bringing the amino acid on time to this conveyor belt. Okay? Yeah, I'm not going to hold on. tRNA, transfer RNA, and bring the amino acids. This is a big picture in terms of proteins. Gene, transcription, we get an mRNA, translation of the mRNA, we get our polymerase amino acid sequence, we get the polymerase. Where do you think this is happening? The nucleus is transcription. Okay? While is the translation also in the nucleus? In the cytoplasm or roughly, that's going to be outside. So this transcription is in the nucleus because we have our genes, we have our DNA in our nucleus. Transcription is the nucleus, while translation will be in the cytoplasm. And this is what I mentioned about what structure allows mRNA produced in our nucleus to come out into the cytoplasm. So what allows? No, centrosomes. We'll talk about centrosomes someday. What allows our mRNA that was produced in our nucleus to come out of the nucleus?
So nuclear envelope.
Nuclear envelope, and what structure in the nuclear envelope was it? The nuclear pore, because mRNA is a big molecule. Okay? Not as big as DNA, but it's still a big molecule. Okay? It comes through the nuclear pore, the larger things in our nucleus. Okay, so translation happens in our cytoplasm, transcription happens in our nucleus. So next we're going to talk about cell cycle and cell division. It's called a cycle. I always think about a circle, right? It's called a cycle because a newly formed cell undergoing lots of changes before it really splits into two cells. And that's what's a cycle. Okay? We can talk about cell division in two places and two kinds rather, not two places but two kinds. Mitosis, where we're replacing old cells, damaged cells, right? This happens everywhere. It even happens in other space. But we're basically producing clones of ourselves, right? While meiosis is only in the gonads, only in testes, only in ovaries. What is different about this cell division, besides what final cells name-wise, but what's different about this cell division compared to mitosis? How many? Yeah, it only will have half the number of chromosomes, right? So it's a 46 chromosomes, we only have 10. While here, we want to keep the 46. We want to have the same DNA, same number of chromosomes, same number of chromatin division, more correctly rather, same number in each cell. Okay? So, we can talk about this cell cycle as interphase and mitosis. Interphase is basically preparation to divide. Okay. Just imagine if I kept carrying a piece of paper, right? Each time I tore it, if I did it equally still, what will happen to the size of the pieces? It's going to get half and half and half, right? We don't want the cell size to be decreased, okay? So we need to increase it first, and then when we split it, we're hoping that it comes to the same size. We want it to come to the same original size, okay? That interphase is that preparation to getting it larger, not only the size, the number of organelles, number of DNA molecules, because we don't want to cut in the DNA either. Yes, we don't want the number of DNA, then are we going to cut each DNA into half? That won't be good either, right? So we have to replicate the DNA, then we can separate them out. So we still have 46 DNA molecules. That's the basis of interphase, preparing for the division. And then we'll talk about mitosis, where we're basically separating out the nuclear contents, where we want exactly the same number of DNA molecules, same genes in the cells. Okay. And we'll talk about them as PMAT, the first first letters of the four: prophase, metaphase, anaphase, and telophase, and sequence. We'll talk about some things happening in each stage. Some stages, like the prophase, we need more detail. We'll talk about it. So, let's first talk about interphase. Interphase has three phases. So, I'm going to draw. First of all, interphase. So, newly formed cell, all the stuff it goes through before it's ready to work. So, this black would be interphase. That has three distinct phases within it. Okay? G1, then S phase. And then it's G2 phase. So G1 is where we have the most growth. The cell is growing, organelles are doubling. Okay? S phase is also important. S, you have to think of it as synthesis phase. What is being synthesized is our DNA molecules. We are forming not 46 DNA molecules, but 92 DNA molecules. At the end of this, we will have 92 DNA molecules. Okay? And then we finish interphase, we finish cell growth. The biggest one I always point out here is that organelle. We had one off, which I mentioned in our lab, that it helps to separate the chromosomes. What was that? With black, with two yellow centrosomes. So that one we need two of, because we want the two to sit on two sides of the cell. They're going to try and separate the chromosomes out. Okay, so we achieve two during G2 phase. Okay, that's the key in that. Okay, so in G1, lots of growth, organelles are doubling. The amount of cytosol is also increasing rapidly. S phase is next. DNA replication occurs here because we want to be able to separate out two separate nucleus with identical DNAs. Okay, number as well as size and information. Okay, and then G2, we're continuing the cell growth. One important organelle that doubles here is the centrosome. Because we need two centrosomes during mitosis. Meiosis is happening elsewhere. Similar kind of method. But during mitosis, we're actually going to separate out the nuclear contents. Okay? So here, this would be part of mitosis. now it's almost doubled in size.
Wait, so the two centrosomes is to duplicate the
So the nuclear content, so when we talk about chromosomes, we want to be able to separate them. If you have only one centrosome, you can only grab one, the other will remain in the middle. So we have two, so we can grab. It's like having two Fisher persons on either side, trying to grab the chromosomes and separate them. So we need two of them. At the end of G2 phase, we have two. By the way, when I mentioned there are 96 DNA molecules, each chromatin actually has two DNA molecules. It's not that we're going to have 96 chromosomes or chromatins. We still have 46, but each one has two because we want to keep them together, right? Because if we just let these 96 just float or 92 float around and we've been dividing, how do we know which one needs to go on what side, right? We want the exact same DNA to go on that side. So each chromosome will have two identical DNA molecules. Okay? We'll talk about that. We'll see how they look like here. So here's where I wanted to explain the difference we already talked about. The difference between meiosis and mitosis. Meiosis is the formation of gametes, formation of sperm, formation of oocyte. The big difference is the halving of chromosomes, the number of chromosomes. That way, when fertilization occurs, we go back to the 46 rather than doubling each generation. And then we'll also talk about what else happens to it besides just separating parts. Okay? This is meiosis and mitosis. While mitosis, let's talk about the phases. So it's the PMAT. So prophase, I would suggest, has three important things happening in prophase. Summarize what's called there. Prophase. What was it? What are microtubules? Yeah, they're tubes, they're hollow tubes, they are part of our skeleton, cytoskeleton they're called, right? We're going to talk about how the centrosome is going to produce that fishing line that's going to try and grab onto our chromosomes. So first and foremost, if we have a nice dark nucleus, is it going to be easy to divide? No, right? You can take a meat cleaver and try to cut them, but then it won't be equal in terms of unit. Okay? So what we're going to do first, or some of them is sequential. These are really happening almost at the same time, just slightly different timing. So the nuclear envelope fragments. And nucleolus disappears. Then you have the chromatin, which was the less condensed form of DNA, condensing further. Condensing means it's packing up. Okay? Like if you were to move out of house, how easy would it be to take one book, one mug, one plate at a time? Right? That's not going to be so easy, right? You'll put it in a box, it'll be easy to carry out. That's kind of what's going to happen here. We're going to pack it nicely so we can separate and then carry it away nicely as well. Okay? So chromatin condenses down, condenses to form chromosome. Chromosome is the highly condensed form of DNA. Okay, so chromosome is the highly condensed form of DNA. Third thing that's going to happen is our centrosome. Form centrosomes, there's two, form So this one has multiple names called mitotic spindles, spindle fibers, microtubules. They're all same. They're just describing. One is describing structure. The other two, well, two are describing structure, mitotic spindles. The other one is talking about how they will do. Okay, so if you see in different ways, you write them different or you read them different, it's the same thing. Okay, form mitotic spindles. Mitotic spindle fibers, let's say mitotic fibers, since that's what's on the slide. Okay, and we had two, they would move to opposite poles. Let's go attach chromosomes, which one's moving back? Let's put the attach and do that again. And move. Next phase is the meta phase. This is very distinct phase. What we want is the chromosomes aligning equator of the cell. So what is the equator? The middle, right? So it's the equator, it's an imaginary middle plane. Okay. So at this time we will have the chromosomes aligned at equator with mitotic spindles or spindle fibers, mitotic fibers attached. Because if they're not attached, we can't separate them. Okay? So in metaphase, they're lined up nicely at this imaginary middle equatorial plane. If the two centrosomes are doing this way, the two centrosomes are the other north and south. So all the chromosomes are in the equator. With them attached. So next phase will actually break and then okay. And here I want to emphasize about chromatid. I will drawing on the next slide. So this is metaphase. The chromosomes are aligned at the equator with spindle fibers attached to it. Okay. Next phase. Here we see the chromosomes looks almost like an X almost because it's got an H even. Right? Because each one of this, what's the way to say, the runs with the sides, if you will, has a DNA molecule. Okay, this is one chromosome with two DNA molecules. Okay, so I'll call this DNA 2. Orange one's DNA 1. This is one chromosome. Each of this is actually called a chromatid, the D. This would be chromatid 1, containing one DNA molecule. This would be chromatid 2. Okay? So these are lined up at the equator, say, in that way, it should run this way. This way, the two chromosomes are going to be on either side. They're going to be attached to this chromosome on either side of the chromosome. What's going to happen in the next phase is two things. First, we're going to split this guy up. Okay? When we split it, we'll still have identical DNA molecules going towards the other side. So anaphase has two steps, very important. Please don't only write down one, write down both. chromosomes are split. You can put chromatids if you wish. Chromosomes are split and pulled to opposite poles. So this one, this red, orange will end up on this side, which will become a cell of its own with all the other 45 chromosomes. This one will go to that side, all to the 45, becoming a new cell. Okay. Anaphase, we will separate out the chromatids, we're splitting the chromosome, and we're moving. That's why here it looks like a V, with the two H's got split. They're pulling by the middle, so they'll look like these. Okay? So these two will attach to each other, these two reds will attach to each other, these two blacks will attach to these two reds. They will pull, they will split the chromosomes and then pull them to the opposite poles. We will end up with 46 chromosomes, with 40, 60 inside. Telophase is kind of the opposite. We've already separated out. Now we're going to go back to forming nuclei. Really, it will be two nuclei at one time. So basically, opposite of prophase is telophase. So the spindle fibers disappear. Mitotic spindles disappear. Mitotic fibers. As a chromosome becomes less condensed. Chromatin again, and nucleolus deforms, and nuclear envelope forms. Material forming them. So at one time you're actually going to have two nuclei for a brief period of time, after which we will separate the two cells separately. Which is the cytokinesis we talked about later. Cytokinesis is the division of cytoplasm. So it's actually already started here, really starts at late anaphase. As soon as we start separating the nuclear contents, we don't have to wait for two nuclei to form, we start splitting the nucleus out, to split the cell out. So it's the division of the cytoplasm. It starts really in anaphase. What happens is like, imagine if we have a balloon, okay? We put a little drawstring in the middle and start tightening. Okay, that's what happens. Contract by the ring, it just tightens, and it splits the two cells. Okay. Sometimes called a cleavage or a cleave. You cut, furrow, dip. Right? See the cleavage, furrow. Basically, cytokinesis is the division of cytoplasm, and it ends up with two separate cells. Any questions there? So, Let's talk about cell division in cancer. We mentioned that cancer is basically an uncontrolled cell division. Okay? Why does it come about? It's because of mutations that may occur in the genome. Do mutations happen often? They do. Okay? Do we fix them most times? We have enzymes that can fix them as we go on. But it can result in cancer cells. So again, we have mutations that happen, nothing much comes off of it. They're destroyed before they divide, or even after they divide, they can be found out by our immune system. Okay? So cancer is basically the cell division and growth exceeds normal cell death, so it just starts to increase in size. Whereas, before that, what's the difference between benign and malignant? I started talking about it, but I want to read. So the basic definition, yes, we may say malignant is more dangerous, true, but benign can be as well. Okay? Yeah, we'll talk about metastasis. That's even going further. Okay, so benign stays within the same tissue. Okay, while malignant spreads throughout. First starts with the same organ, spreads to the other tissues. Then, if it spreads to other organs outside of its vicinity, that's metastasis. Okay, now metastasis is when it goes from one organ to a different organ. Okay, while malignant, it just grows through other tissues. Benign tumor, I'll tell you about a dangerous benign tumor. Meningioma. Where's meninges? Where are meninges? Yeah, around our brain and spinal cord. It's a little covering around. Meningioma is actually a benign tumor. It stays within the meninges. But why is it dangerous? Yeah, it's going to squeeze. If you're not growing to this, skull will just push back. But it grows inwards, right? It's pushing on to our brain. That's why it's dangerous. Yes, most often it's more dangerous than benign, but it's not exclusive. And it's not a definition of it. Benign just means it stays in that tissue. Malignant goes beyond that tissue. Metastasis goes beyond that organ. It goes to a different organ. Right? Breast cancers can metastasize to lymph nodes, liver. Right? Lung cancers can metastasize to bone, for example. Prostate cancer, without bone, just goes to bone. And that's metastasis when it goes outside of that organ. Whereas we do have preventing, right? As I mentioned, DNA mutations do occur often because cell division is happening all the time, which means DNA replication is happening. And we do have some enzymes that can be replicated. And if it goes beyond, we also have apoptosis. That's programmed cell death. We recognize the cell to be non-functional, potentially cancerous. Our immune system can attack. Okay? But a cancer tries to go around all these controls. Yes, but the definition of benign tumors confined to that tissue, right? Whereas malignant tumor, they go to surrounding tissue. Okay? And then when they go to other organs, it's metastasis. Questions? Let's take a little break and then we'll go to tissues.